For a general chemical reaction at equilibrium, represented here, the concentrations of the species on the two sides are related to each other by an equilibrium constant, K, and the expression for that equilibrium constant is familiarly written this way, where the capital letters in brackets represent the concentrations of the species involved, and the lowercase letters represent the stoichiometric coefficients. We're usually interested in what drives a reaction in a particular direction and gives us a favorable equilibrium. And ultimately, the magnitude of the equilibrium constant is determined by delta G, what's known as the Gibbs free energy change for the reaction. And that equation relates the change in free energy to minus RT log K, where R is the gas constant, T is absolute temperature, and K is the equilibrium constant. The Gibbs free energy change is a relationship between the relative stabilities of the reactants and the products. For a reaction where the starting material is less stable than the product, when we have a decrease in free energy, so a negative delta G, and that's referred to as an exergonic reaction, and it's said to be thermodynamically favored. Now, you might be familiar with this same sort of explanation related to the heat of a reaction or the enthalpy change, and that would be referred to as an exothermic reaction. If the reaction gives rise to a product that is less stable than the starting material, then there would be an increase in the Gibbs free energy and a positive delta G, and this is what's known as an endergonic reaction. If you limit yourself to being concerned with the enthalpy change in the reaction, that would be an endothermic reaction. In general, we find that more stable products are favored. A negative delta G means that we have a favorable equilibrium constant for our reaction. So as I stated, more stable products are favored in an equilibrium. But where does that equilibrium lie for a given change in free energy? And we can see that data here. And one of the conclusions we can see from this data is that often very small differences in the enthalpies or the free energies of the reactants and products can give rise to reactions going nearly to completion. So for example, if you draw your attention to the middle of the chart here, you can see that just 1 kilocalorie difference per mole between the reactants and products gives rise to an equilibrium constant of about 5.5, meaning the reaction goes to about 85% completion. And if we extend that down, you can see that when you get to about 3 kilocalories per mole, you have a reaction that's essentially complete, over 99% of product being formed at equilibrium. Some reactions proceed in a series of steps, but still may be thermodynamically favorable as long as the starting material and the final product have a negative delta G of sufficient magnitude. As long as any intermediate energy barriers can be overcome, the reaction should proceed. If the barrier to the formation of the intermediate only generates a small amount of it, that may be a little consequence because it could react rapidly to move on to the product. And as Le Chatelier's principle tells us, more of that intermediate then would be formed to reestablish equilibrium to that intermediate step. If the Gibbs free energy change ultimately determines whether a reaction is thermodynamically favored or not, it's useful to break that down and look at what contributes to the free energy change of a reaction. And we find that it's composed of two things: the enthalpy change of a reaction and the entropy change of a reaction. And in the relationship, we can see that the entropy change is temperature dependent. That this term is the product of the absolute temperature times the change in entropy. Looking at these two terms more closely, the change in enthalpy is determined largely by bond strengths. If the products have stronger bonds than the reactants, then the enthalpy change will be a negative number. And that's a favorable value because a negative delta H would contribute to delta G being more negative and thus more favorable. The entropy term, on the other hand, is more related to freedom of movement. If the products, for instance, are less restricted or ordered than the reactants, then there's an increase in entropy, and that would favor the delta G being more negative because of the minus sign in front of the T delta S term. You can also see that the delta S term becomes more important as the temperature rises because the magnitude of that product would increase and thus make delta G more negative because of the negative sign. There are ways of qualitatively assessing whether there would be a positive entropy change or a negative entropy change. Generally speaking, if you have a reaction that goes from 1 mole of reactant to 2 moles of products, that's going to increase the freedom of movement because you're generating more particles from fewer particles, and you would have a favorable delta S for that reaction going forward. As I stated, enthalpy change is related to bond strengths. Going from reactants to products, you're typically breaking and making bonds in the overall process. And if you are collectively breaking weaker bonds and making stronger bonds, you can make a good estimate of the enthalpy change of a reaction by using bond dissociation energies such as those listed here. And that's an exercise you probably did in general chemistry where you calculated an estimate of delta H by simply looking at bond dissociation energies for the bonds involved in the overall reaction. Well, the change in delta G determines whether a reaction is thermodynamically favored. It doesn't tell us anything about the rate at which the reaction proceeds. That's determined by the height of an energy barrier that separates the starting material from the product. And in fact, we find many examples, even in nature, of unstable starting materials that are protected from transforming into products by a very large kinetic barrier. In any sample of molecules, they exist in a distribution of energies known as a Boltzmann distribution, and they typically have the form of a bell curve of sorts. And these represent the average energy of the molecules in a sample of molecules. If you raise the temperature of the molecules, usually by applying heat, you simply change the average of this distribution of temperatures. There's always a small number of molecules that have a sufficient amount of energy to overcome an activation barrier for any given reaction. But if the number is too low, then the rate of reaction may not proceed at an observable rate. Sometimes we can get to an observable rate simply by warming the reaction mixture. It's also a rule of thumb that if you increase the temperature of a reaction by about 10 degrees Celsius, you'll roughly double the rate of the reaction. And that's occurring because we're increasing the number of molecules that have the energy necessary to surmount the activation barrier. The rates of reactions also depend upon the concentration of the reactants. For some reactions, that concentration dependent only depends on one component. There may be a single reactant that's transformed into products. And we can express the overall rate of that reaction as a constant times the concentration of that reactant. The constant, K, is known as a rate constant, and it's a fundamental property of a given reaction. It determines how that reaction's rate depends on the concentration. The units of that rate constant are dependent on the form of the equation. And you can see in this equation, we typically express rates in terms of molarity per second. And so the rate constant would have the corresponding units to make the rate come out with the proper units. Other reactions depend on the concentrations of two components of the reaction, so-called second-order reaction. And they have rate equations that look like this, where the rate is dependent on the concentrations of two components of the reaction. Correspondingly, the rate constant has different units to derive the rate as molarity per second. As I mentioned previously, exothermic reactions may be very slow, indeed immeasurably slow. And it's a good thing because we're surrounded by many things in our world that are in fact combustible, and we are walking around in a sea of oxygen. And fortunately, there's a significant barrier to even very favorable reactions. Here's the reaction that takes place when we burn natural gas, which is the combination of methane and oxygen producing carbon dioxide and water. You probably know that if you turn the gas on in a gas stove or a Bunsen burner, it doesn't just spontaneously combust. You have to supply some energy in order to get the reaction started in the form typically of a spark or a lit match. Now, once the reaction does begin, there's enough energy released in the process that it can supply the energy necessary to the remaining molecules for them to react. So as we go forward from here, we're largely going to talk about first order and second order reactions. In fact, most organic reactions are either that, first order or second order. We rarely see higher order reactions in organic chemistry. And in this chapter, we're going to be talking about substitutions that have different kinetic behavior. We've already discussed reactions that we're going to call SN2. It stands for substitution nucleophilic bimolecular. And those reactions have a rate dependency that depends on both the rate of the substrate and the nucleophile. We're going to see later that there are other types of substitutions that are first order reactions, where the rate of the reaction is only dependent on one component in the reactants, the substrate. So as I stated, even a thermodynamically favorable reaction can be unobservably slow if the barrier to the reaction, which is called the activation free energy, delta G with a dagger on it, is very high. Combustion is one of those sorts of reactions where this barrier prevents methane and oxygen from spontaneously combusting. So that delta G is the step with the highest barrier to the reaction, and it limits the overall rate. And so it's called the rate determining step. And this is the activation energy for the reaction. Now let's look at one of the substitution reactions that we've seen previously. And we can see in this reaction, we have a nucleophile in the form of a cyanide ion interacting with methyl iodide in this reaction. And this is the mechanistic pathway. The pair of electrons from carbon is acting as a nucleophile. It's interacting with the sigma star orbital of the carbon-iodine bond, causing that bond to rupture, and iodine is our leaving group. So here are those electrons that were involved. We formed a new carbon-carbon bond, and we have a new non-bonded pair of electrons on iodine. So what is it that creates the energy barrier? In this case, it turns out to be both a combination of enthalpy and entropy. It turns out there's a very specific requirement that this nucleophile attack the carbon kind of as it's shown from this side of the molecule. There's a direction that it must approach. It can't approach from this side or over here or from there. It has to come in from what's called the backside of the carbon-iodine bond. And that is an entropic requirement that contributes to this delta G being relatively high. But it is a barrier that can easily be overcome, and by simply mixing cyanide with methyl iodide, you actually get a fairly rapid reaction to form the products. It's worth noting that the way we observe reactions and the way we draw them is somewhat arbitrary. They are fundamentally equilibria. The question is, which direction does the equilibrium lie? And we often draw our reactions kind of in the direction we think that the reaction will be favored, but we could draw it the other way. But when we think about the reverse reaction, and here, if we think about this reaction running in the other direction, where the iodide acts as a nucleophile and displaces the cyanide, we can see that that reaction would be exceedingly slow because it needs to climb a very high energy barrier in order to get to the products. So regardless of the reaction being endergonic and disfavored thermodynamically, it's also highly disfavored from a kinetic point of view and would be even slower than the forward reaction. Now, it's worth mentioning whether a reaction would actually follow the same pathway in the forward and reverse sense. But we know from something known as the principle of microscopic reversibility that this is what indeed will happen. And the notion is this, that imagine that you have two villages on the either side of a mountain range, and there are a couple passes over that mountain range that have roads along them. And the difference between these roads is the height of this barrier that you have to climb over the pass. And the principle of microscopic reversibility says that the low energy pathway in one direction is also the low energy pathway in the other direction. And so the forward and reverse reactions are actually going to favor the same pathway, even though there are multiple pathways available.